All right, thank you, Dan, for the kind words. So today I'll be talking to you about uses of genetic data in psychology. Hold on. Okay. So it turns out that psychology already has a subfield that has, for a long time, made extensive use of genetic data. This subfield is called behavioral genetics. And its practitioners study the inheritance of mental abilities, personality, vocational and recreational interests, religiosity, political beliefs, as we were just hearing about, mental illnesses such as schizophrenia and autism, and also fertility, which was recognized by Ronald Fisher more than 80 years ago to be in human beings as much of a behavioral trait as a physiological one. Now, talk about abilities and personality and so forth is fairly coarse-grained. At a finer grain, we might say that behavioral genetics aspires to address how the genome encodes a brain capable of more elemental cognitive feats, such as seeing, thinking, talking, and so on. So I'm going to start out by giving some motivation, providing some reasons why I think behavioral genetics is a scientifically worthwhile endeavor. So I'll start with the observation that a typical human brain consists of 100 billion neurons. That is, cells that are specialized for information processing, and at least 10 trillion connections between these cells, which are called synapses. I say at least because these things, even now, are still difficult to count, and so the number of synapses in the typical human brain may be orders of magnitude larger than this. So anyway, these are very large numbers that we're talking about. In fact, this number of neurons is roughly equal to the number of stars in the Milky Way galaxy, and their interactions with each other are much more complex than the mutual gravitational influences that give the galaxy its overall structure. Now, interestingly, all this complexity flows from a genome consisting of only approximately 100 megabytes. Now, that precise number depends on certain assumptions, such as the fraction of the genome that's functional, things that we don't know for sure yet. But nevertheless, that number is in the ballpark. And to many people, it's a surprisingly small number. I mean, for example, on my computer here right now, I have several GWAS meta-analysis output files that are each bigger than 100 megabytes. And so we have this disparity in magnitudes that leads to an interesting question. How is it that such sparse data is capable of encoding a device of such enormous complexity? Another motivation for studying behavioral genetics is the so-called nature-nurture issue, a euphonious name for this opposition that was coined in the 16th century. These particular lines come from Shakespeare's play The Tempest. They're spoken by Prospero, the main character, when he's asked to explain the character of the villain Caliban. Interestingly, later in the play, when Shakespeare gives Caliban a chance to speak for himself, he has a somewhat different opinion. But regardless of who's right, this is a question that's older than science itself, considered by Aristotle and Plato, and surely pondered by at least some people even before the time of the ancient Greeks. Nowadays, we might put the question this way. How is it that biological endowment interacts with experience and culture to produce behavior? Okay, now this next motivation focuses on qualitative properties of the human mind rather than individual differences. So we have this school of thought in cognitive science called nativism, which posits special capacities, sometimes uniquely human, sometimes shared with other animals, that are called, depending on the theorist, modules, faculties, or course systems. So perhaps the most famous example is Noam Chomsky's innate universal grammar, which has been called by others the language acquisition device or the language instinct. Now, a relevant question which used to be entirely neglected but seems to be attracting more attention is whether it's possible for the genome, any genome, to encode capacities of the kind that are hypothesized, and if so, whether genetic evidence can be brought to bear on these hypotheses, and thereby give the discussion less of the armchair philosophical character that it has traditionally assumed. Finally, behavioral genetics is important because of its connection to the study of human evolution. In fact, evolution can even be defined as the changes over time in the frequencies of alleles. That is, classes of types of genetic material, such as, for example, the famous GCTA when we're talking about single nucleotides. Now, the subfield of evolutionary psychology treats human nature, by which is meant not only cognitive capacities such as language, but also, for example, our sexual and moral instincts, and also social behavior such as religion, as products of biological evolution. And to me, at least, it's very obvious that this approach can only benefit from the deciphering of natural selection and other evolutionary forces in modern genetic data. So I've laid out many promising connections of behavioral genetics to other parts of psychology and science at large. Despite these connections, however, behavioral genetics has traditionally been a somewhat isolated and insular field. We can see this in the fact that the vast majority of psychology departments still don't have a behavioral genetics program. This is something that really goes on only in a handful of places. But maybe that's okay. Maybe that just means more glory for us. But I think that the neglect is worrisome to the extent that it goes in both directions, such as when behavioral geneticists, sometimes almost with pride, admit that they don't know much about the brain or cognitive psychology or what have you. So given this historical situation, whatever its causes, you could have a whole lecture about the causes just by themselves. But given this situation, we have to ask ourselves the question, is it worthwhile to struggle against the evident inertia and make genetics a more integrated part of conceptually neighboring intellectual disciplines? Now I'm going to show you a video of some twins separated at birth who participated in a study conducted in my own department by my predecessor Tom Bouchard. This is the Minnesota study of twins reared apart, or MISTRA for short. Now my own PhD advisor, Steve Pinker, was interested in questions of evolution and human cognition at the time that the paper reporting the MISTRA results came out in Science back in 1990. And at that time, he had never heard of behavioral genetics. But when he learned of these results, he was so impressed, so blown away, that since then he has permanently incorporated behavioral genetics into his own thinking. So let's watch this video. The psychologist you'll see is Nancy Segal, who was a postdoc at Minnesota while the study was being carried out. She's now a professor at Cal State Fullerton.
All this made us wonder, what makes us who we are? Is it how we grew up, or is DNA our destiny?
Hi, Tamara.
Researcher Nancy Segal has spent a lifetime trying to answer these questions. She has interviewed more than 50 pairs of identical twins who were separated at birth.
Identical twins raised apart are exceedingly rare. They're very, very valuable cases to scientists. We're very interested.
She wants to know how Tamara and Adriana's similarities compare with other identical twins separated at birth. What can they teach us about why we behave the way we do? Why we pick certain jobs, certain friends, even certain shampoo?
To the extent that identical twins are more alike in these things, we can say with a fairly strong degree of confidence that yes, genes do play a role.
48 Hours found 4 sets of these scientific gems and brought them to New York.
All 4 of you, 4 pair, have had a similar experience. You were identical twins, separated at birth, adopted and raised by different kinds of families, and then came together as adults.
Identical twins Tom Patterson and Steve Tazumi met just 4 years ago. Tom is from rural Kansas. Steve lives in urban Philadelphia. Their upbringings couldn't have been more different.
I was a Christian and he's raised a Buddhist. His dad was a pharmacist and my dad and mom were janitors.
Still, Tom and Steve chose the same careers.
It's phenomenal is that he owned a bodybuilding gym and I owned a bodybuilding gym. And so we were both definitely 100% into fitness.
Yes.
Daphne Goodship and Barbara Herbert first met when they were 40. They too discovered remarkably similar life experiences.
We discovered we'd had a miscarriage the same year, followed by 2 boys and a girl in that order. We've even had cooked the same meal from the same recipe book.
On the same day?
Yes.
Without knowing?
Without knowing. We've done it a few times, but that one we can remember clearly.
You were raised
Jewish.
and you were raised...
I was raised Catholic and now I'm Christian.
Debbie Melman and Sharon Posette were brought up in different faiths with different traditions,
Fill that void.
but their habits are incredibly similar.
When you get excited, you
We
cross
cross our eyes.
You do too?
Yes.
Oh my God.
And our dads would have said the same, like, don't you roll your eyes at me, young lady.
Of all these twins, Roger Brooks and Tony Malassi have the most dramatically different backgrounds.
Well, I was in an orphanage home the first 5 years.
While Roger bounced from home to home, Tony was adopted by a loving family.
I had a very secure, good childhood, a lot of love in the family.
Do you see a big difference because he didn't have that early stability?
No, I don't. I didn't live in a traumatic environment. I lived in a good environment. You had rickets? No. No, he did. His shoes were tight. And he doesn't realize how tough he had it.
Having gone through all of those difficult times, Roger is still an upbeat, positive person.
Still Tony.
Yes. I think it speaks to the genetic predisposition to just simply have a sunny, upbeat, happy personality.
Research shows that identical twins raised apart are more alike than they are different, and they're surprisingly more alike than the brothers and sisters they are raised with. We asked Nancy Segal to design an experiment to show how genes play a role in our everyday choices.
We were interested in their choice of places to go on vacation and what kind of foods they might choose given an array of choices.
Ribs.
How they moved and gestured. We'd like you to stand on one foot. Barbara and Daphne are called the Giggle Twins, and I think you can understand why.
They can't see each other. This is a
No,
partition.
that's a partition. They can't see each other. However's most comfortable.
Oh, look at that. Oh.
Now, they're
And
not,
they're doing this
they're
without
not
seeing
sure which
each
is
other.
more comfortable. And
But they keep doing it exactly
they're not certain which is more comfortable. Now we've asked them to dance. We want to see how their bodies move. And they show
look
a
at
lot of
that.
similarities. See how they're moving?
Look at that.
There they go.
Oh, look at that. Oh, my.
See, and here they can't see each other, but they're moving the same way.
I just want everybody to pause. You're sitting there like this. Get this picture. And you're sitting there like that. Your legs are crossed. Can you see this? I can't see what you guys are doing back there. Your hands are the same? On the knees, both of you?
They're the same
Oh,
thing.
my God. Look at this. What do you make of the weird coincidences?
I wouldn't call them weird coincidences because I think they're scientific phenomena. If you think that all of us are more comfortable in certain ways because of the ways our bodies are made, and if all of us had identical twins, they'd be sitting exactly as we are.
You're saying that somebody would cross their hand this way because their body is made in such a way that this just is the most comfortable way
Exactly.
to sit.
Siegel believes that's why Adriana and Tamara dance the way they do.
One could say, well, their mothers gave them dancing lessons. But the question is, why did their mothers give them dancing lessons? Because the girls were agile, enjoyed it, did well at it.
Adriana and Tamara, like these separated at birth twins, believe they are in fact more identical because they were not raised together.
You don't have that baggage of, oh, mom, like, I mean, I'm sure identical twins
like,
had
you
that.
know,
You stole my boyfriend, or you studied and you got a 92 and I got an 88, or something, you know.
Because they never had to compete with each other, they believe they were freer to express their genetic potential.
You are what you are.
You're genetically
Yeah, I think
packaged.
so, yeah.
So does it...
Wow. Still get the chills every time I see that. So, a little personal story. I actually first learned about the Minnesota study of twins reared apart from, at the time, my future PhD advisor, Steve Pinker, through his book, The Blank Slate. Now, it's difficult to say for certainty that, reading this book or meeting that person or what have you actually changed your life, because as we've just seen, the puppet masters pulling on your strings are not necessarily what they seem to be. But I'm willing to go out on a limb and say that Steve and also Tom, Nancy, my current colleague Matt McGew, the whole Minnesota gang, really did change my life. Because of Mistra, I'm standing here in front of you today talking about this stuff instead of, I don't know, working for some law firm in New York. So this is why so many of us find this so worthwhile. When there's something like this out there, waiting to be understood at so many levels, then as a scientist, you feel very lucky to know about it and to be hot on the trail. Okay, now I do want to add a qualification. So these remarkable similarities, such as both twins owning a bodybuilding gym or both twins marrying women with the same name and giving their children the same names, these are just about never observed in studies of other kinships. So this is almost certainly because these very specific particular outcomes are the result of epistasis, which is just a fancy genetic term for nonlinear interactions between different sites in the genome. This is discussed in one of the assigned readings, although for some reason the authors, as academics are wont to do, made up their own term, emergenesis. But what they really mean is just epistasis. And so, for many reasons, including the combinatorial explosion when you consider the millions of polymorphic sites in the genome, despite the enormous ongoing progress in modern genetics, the intricacies of epistasis will probably continue to elude us for quite some time. So today, I'm going to talk about something that actually may be tractable when tackled with modern genetic data, and this is the riddle of sexual reproduction. So certainly, sexuality and sex differences are popular topics in psychology. In fact, evolutionary psychology has been called derisively the psychology of mating. And certainly in my own experience, it seems that younger people who are attracted to this field seem to be of the sort of sketchy type. But setting that aside, well, not the elderly senior citizens like David Geary, but anyway, the point I want to make is that these surface phenomena overlie a deeper and more fundamental issue. So before asking questions such as why is it that girls tend to get higher grades in school than boys, or why in our prisons we have more men than women, we can ask actually a more fundamental question than even these, and which is why is it that we even have two distinct sexes at all? For example, you can imagine that when it comes time to reproduce, you would be able to find as a partner any member of your species instead of being restricted to a special 50%. So why is it that we even have this restriction? It seems like we would be fine without it. Furthermore, to go even deeper than that, the complex process of sex itself, featuring male and female specializations, even this does not seem to be logically necessary. So why not reproduce asexually, as many species continue to do? That is, once you've gotten tenure and you decide now it's time to start a family, why don't you just like butt off a little spore, just like C. cerevisiae are able to do. And so the famous biologist William Hamilton put it this way: sex is an intellectual problem for some of us, but a personal problem for us all. Okay, so to illustrate the complexities, here's this toy picture of the genome that I included in my class notes on genetics. So indeed, there are all these molecular complexities that ensure that there's a reduction from diploidy to haploidy, where contributions of your own two parents are stitched together, and then a reconstitution of diploidy from the haploid state in this process that is called mating. And so the question is, we have all this elaborate molecular machinery that supports this complex process, all these psychological adaptations that help us with this, but why do we have all these things when it seems that this alternative would do just as well? Now, besides the fact that we have all these molecular complications that it's hard to understand why they're there, there's also a cost of sexual reproduction that's a little bit easier to understand, I think. So to understand this cost, let's suppose that we have two very similar populations, one that reproduces sexually, the other reproducing asexually. And what does similar mean? Similar means that these two populations are basically competing for the same finite niche in nature. So it's a zero-sum game. The success of one tends to reduce the success of the other. Now, every member of an asexual population is essentially female. Now, let's suppose that reproduction here is limited by the amount of resources that each female can bring to reproduction, so it's not a density limitation or anything like that. If each such female is capable of bearing two offspring, then it's obviously the case that the asexual population will double in size each generation. So we start with 1, and we go to 2, and to 4, and so on. Since only half of the equivalent sexual population gives birth to children, the same birth rate, 2 children per female, keeps the sexual population at a constant size. So naively, you would expect that if these two populations are both competing to fill up the same slots in nature, that the sexual population should quickly swamp the asexual one. And this apparent disadvantage that sexual populations labor under has been called the twofold cost of males. That is, why is it that we have all these worthless males in our own populations? Now, this cost is even greater than implied by the preceding argument. This is because mating can be dangerous, time-consuming, and energy-draining. The sexual act itself can transmit diseases to the partner or the resulting offspring. And also, when you have sexual reproduction, this almost inevitably leads to sexual selection, which leads to traits in males such as greater size, weapons, and so on. From the fact that these are not present in the females, we can infer that these are not needed for basic survival. And so therefore, the investment that you need to grow these things should be counted as a cost. Perhaps the greatest cost is the possibility of failing to produce any offspring at all. It's often the case that only a small fraction of the males father the entirety of the next generation. So it might seem naively that if you're one of these unskilled or unlucky males, that it would be kind of a good idea for you to at least have the option of reproducing asexually. There are some species that are like that, where the mode of reproduction is facultative. You can either go sexual or asexual, depending on the circumstances. But in most vertebrates, say, that option has been foreclosed. It is obligatory to reproduce sexually. So apparently, natural selection, even if you're one of these male walruses who's not big enough and not ferocious enough, that natural selection favors the strategy of continuing to try, no matter how hopeless it seems. Now here's a way to see that sexuality, another way to see that sexuality is what natural selection indeed seems to want. So let's presume that asexuality is the initial state, which is safe to infer because today we see that most asexual species are unicellular organisms that are probably not too different from our common ancestor. So it turns out that there are some vertebrate species, you can find them in various odd places like this lizard in Texas, that do reproduce asexually. That is, all members of this species are female. And so these species are an existence proof that a switch back to asexuality is logically possible. And this is something that, because of the twofold cost, we might expect to just come roaring back if it is not disfavored by natural selection. But the thing is that asexuality does seem to be disfavored. So we can tell from various phylogenetic comparisons that most asexual vertebrates, like this lizard, recently evolved from a sexual precursor. Conversely, long-established asexual animal species are virtually non-existent. So the implication of this is that if a species happens to become asexual, it will be around for a while, but then it will quickly go extinct or revert back to sexuality. Therefore, sex must confer an overwhelming advantage. But what exactly is this advantage? If it's so overwhelming, then why isn't it obvious to us what it is? So this is the problem of sex, the intellectual problem at least. All right. Okay, there's a bit of a delay here. Okay. So there are many different qualitatively distinct families of hypotheses that have been advanced to an attempt to solve this problem. I'm going to talk about just one of these, which the theorists now all agree has at least some truth to it. And more importantly for our purposes, it may be something that GWAS data can help confirm. But to understand this family of hypotheses, we need to define a particular concept called linkage disequilibrium, or LD for short. So what does this mean? So suppose that in the population at large, that the count of fitness-enhancing alleles at one site is negatively correlated with the count at another site. So to make it more concrete, suppose that you inherited 2 good alleles, fitness-enhancing alleles, plus alleles at one particular site, one from mom, one from dad. From the fact that we know that, we can infer from the fact that there's a negative correlation that at the other site in the genome that we're considering, that you tend to have maybe 0 or 1. Okay? When there's a negative correlation like this, we will say that the 2 sites are in negative LD. Okay. So first of all, this is an actual correlation matrix from chromosome 22. The bluer, the lower the correlation. This is a blow-up of just a tiny part of the diagonal. So it's quite clear that, as a rule in the genome, if we're considering 2 SNPs that are far apart, the measured LD is going to be very, very, very small. But keep in mind that for our purposes, what I'm going to talk about now, the magnitudes of the relevant LD coefficients don't have to be large. They could be extremely tiny. In fact, maybe even impossible to sample small. But nevertheless, these could be relevant. If you have a covariance matrix with a dimension 10 million, if you're calculating something like the additive genetic variance, there's a lot of off-diagonal pieces. All right. So for the purposes of the discussion that follows, let's assume for now that negative LD between fitness-affecting sites is the rule in the genome. So these LDs might be really, really tiny, but let's just assume that they're non-zero and that they are negative. So for now, this is just an assumption. Later, I'll explain what reason there might be to think that this actually might be the rule. So negative LD means an excess of plus minus and minus plus haplotypes. That means that we see more of these kinds of haplotypes where you have the fitness enhancing allele here, the fitness decreasing allele here, than you would expect given the product rule for independent events. So in an asexual population, if you are plus minus at these two particular sites that we're thinking about, then it is inevitably the case that your offspring will also be plus minus at these two sites. Okay? That's just how asexual reproduction works. It's like producing a monozygotic twin in some sense. If you are a sexual creature, however, then the situation is quite different. By recombining the genetic contributions of your own sexual parents, you can pass on a plus-plus gamete, that is a sperm or egg cell, to your own offspring. So let's look at this example. So even if neither parent gave you a plus-plus haplotype, suppose everyone in that generation was plus-minus for whatever reason, but by breaking off a piece of your dad's contribution and connecting it to a piece of your mom's contribution, you can pass on a plus-plus haplotype when it comes time to become a parent yourself. So here's a visualization of how this process affects the population statistics. So suppose that for whatever reason, every haplotype in the parental generation is either plus minus or minus plus. Moreover, if the effects of these two variants on fitness are equal, then the heritability of fitness is actually 0 in this population because in fact there are no genetic differences in fitness. The heritability is a ratio of variances, the top being the genetic variance, the bottom being the total trait variance. And here, if everyone is the same, then the additive genetic variance, this distribution is just a pile of probability on one value. But once sex has taken place, once recombination has taken place, from such a parental generation, you can have offspring representing the two haplotypes that were previously absent in the population. Okay? That is, in the offspring, because of recombination, plus and minus are not as disproportionately paired. The plus and minus haplotypes occur at frequencies closer to those predicted by the product rule for independent events. Okay, but what is so good about that process? So why is this particular effect of sex and recombination a desirable one? Well, intuitively, this is because natural selection now has an easier time increasing the frequencies of the plus alleles, or to put it exactly the same way, to decrease the frequencies of the deleterious alleles. Before, natural selection was in fact impossible because everyone was genetically equivalent. That is, if one person survived and reproduced and another person did not, this would have no consequences for the genetic composition of the population because everyone's exactly the same. But after sex and recombination, now natural selection is possible. That is, the plus alleles can now increase in frequency because now it matters whether one person survives and reproduces and another does not. So instead of everyone being plus minus and nothing ever happening, or the population just deteriorating under mutational load, natural selection can now efficiently eliminate minus and allow plus to contribute more to the ancestry of future generations. This insight can be expressed more formally in terms of the breeder's equation, which I've explained here. So the R stands for the evolutionary increase in the mean phenotype. That is how much the average of the trait increases from one generation to the next as a result of selection. So you can imagine that this is an average of milk yield if you're a cattle geneticist practicing artificial selection, or bristle number in flies if you're a Drosophila geneticist. Or this can be fitness itself. So it turns out that this particular increase, whatever the trait happens to be, is a product of two factors. The first is the heritability of the trait, which again is the fraction of the total trait variance that's due to genetic differences. The second factor, this big S here, that's just the trait difference within the parental generation between those who survive and reproduce and those who do not. Although when the trait is fitness itself, you have to do a bit more complicated analysis, and this turns out to be just the total phenotypic variance. Now recall that if we start with negative LD, then the heritability of fitness is consistently higher in sexual populations. And from that, automatically, we can tell from the breeder's equation that the sexual population increases its average fitness, its average ability to survive and reproduce in its environment more rapidly than an equivalent asexual population. And in fact, if there is sufficient genetic variance in fitness, if the heritability of fitness is high enough, maybe because there are a large number of fitness-affecting de novo mutations entering the population each generation, then the advantage enjoyed by sexual populations can be enough to overcome the twofold cost of males. Okay, so that's what I just said. Okay, so that is why sex helps address the problem of negative LD. But then, this has just been an assumption so far that we have negative LD. Why might negative LD be there in the first place? Now, there are several possible mechanisms by which this might come out. Here, I'm only going to examine one, the one that I think is the easiest to explain. Okay, so often when I'm explaining abstract concepts to my class, I'll do it via these quizzes that I have in class where the students can respond with clickers. After the initial response, they can discuss it and try to change each other's minds. I'm not going to do that here, so instead I'll just read you the question. So the correlation between the SAT verbal and math sections is quite strong, about 0.7 scores and blah, 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 blah. Now suppose that we examine a selected subpopulation where all individuals obtained a combined verbal and math score of 1560 or higher. Okay, so if we're considering just the reading and the math sections, the maximum score is 1600. So what will happen to the sign of the verbal math correlation in this selected group? The options are strongly positive, so it stays roughly the same. Moderately positive, approximately 0, or moderately negative. Okay, so I've given this quiz for 3 straight years now. Nobody has ever given this correct answer ever. An amazing thing when you give an answer question and 70 people in the class all give the wrong answer, and after discussion, again, all give the wrong answer. So I've decided this is not something I should do in the future. But anyway, the correct answer is moderately negative. So here's the reasoning behind that. If we know that a person in the selected group scored 760 on the verbal, then we, for free, know that the person must have scored 800 on the math. Or otherwise, this person would never have made it into the selected subpopulation, and we would never have observed him in the first place. Conversely, knowing that a person scored 760 on math, same thing, we know that they must have scored 800 on verbal. So this reveals sort of a curious property of selection. It can not only change first moments, such as means and so on, it can also change bivariate moments, correlations, and things like this. So in this particular case, the act of selection can turn a positive correlation into a negative one. Now, analogously, perhaps you can already see this by now, the same principle applies to natural selection. So suppose that selection works like this. You have some distribution of the phenotype, and everyone who is 1 standard deviation below the mean or whatever fails to survive or has 0 offspring. So it's like picking the elite population of SAT scores, right? So suppose that individuals who do not carry enough plus alleles in their genomes simply die or fail to reproduce. So among the survivors, the counts of plus at pairs of fitness-affecting sites are now negatively correlated. So if a survivor has a count of 0 at one site, it becomes more likely that the survivor has a count of 1 or 2 at another site. So here's a little visualization of what happens. You have these genomes that are overloaded with these minus genes. Bearers of these genomes tend not to survive. In the meantime, the minus alleles that are left, they tend to be hidden among plus alleles that are sort of hiding them from selection. Okay. So that's, I think, a very simple and elegant explanation of how negative LD can arise. But I hate to spoil things, but it turns out that this particular explanation of negative LD invoking only natural selection is now no longer widely accepted because it's not very general. It only works if epistasis, so remember that concept from earlier, these are nonlinear interactions between different sites in the genome. This only works if epistasis affecting fitness has a certain form. And for various reasons, theorists don't like explanations that turn on something exactly equaling something. So this isn't something that's no longer widely accepted. But there are other explanations, including one that invokes both selection and genetic drift. This is based on the Hill-Robertson effect, which was discovered by Alan Robertson and William Hill. For those of you who are aficionados, William Hill was the PhD supervisor of both Peter Vischer and Naomi Ray. So anyway, he discovered this, and this is a mechanism that provides a viable way that negative LD can arise between different sites. Okay, so these are all ideas that have been developed mathematically over the last, Dan, you have a question?
Yeah, so why do we need negative LD for this whole explanation to work? Because I would have thought...
Well, think about if there are positive LD, then sex and recombination will reduce the heritability of fitness. And then if things started out that way, sex and recombination would actually be a disadvantage.
But I'm thinking of, if you have an asexual population that, let's say there's an initial founder population that doesn't have much variability in it,
Yeah.
alleles, sexual reproduction is going to increase the variability because, you know,
Well, a bottleneck is just like selection, right? So that would be why something like that would cause the population to suddenly exhibit negative LD between different sites.
okay, maybe, but I wouldn't have thought that a founder effect would necessarily, I mean, I can see why selection might induce negative LD. It's not obvious to me why a founder effect would induce negative LD.
It's actually because of the Hill-Robertson effect, which I wasn't planning on explaining here. But anyway, so this is actually an important point. If it were the case that no LD tends to be not distinguishable from zero or actually consistently positive, sex and recombination would actually be bad, because it's like there are some people in the population who have these perfect plus genomes, and now they're just risking throwing that away by engaging in this process.
I would also have variability in the environment that changes what's a plus and what's a minus for fitness.
Oh, so that's an entirely different family of hypotheses. So one of these is the Red Queen hypothesis by William Hamilton. Hamilton was actually a pluralist, so he liked these ideas. He called them the Black Queen. That was his picturesque way of thinking about the effect of mutation, just constantly raining new genomes with bad mutations. And then he called his own theory, which is exactly this: what is fit is constantly changing. The Red Queen hypothesis, after the character in Through the Looking Glass, who tells Alice, here you have to keep running, as hard as you can just to stay in the same place. Okay, so yeah, another question.
Yeah, so why now so this is an elegant explanation, but why do they have to bring genetic drift into the conversation?
Because I mean, so basically this idea that natural selection will cause negative LD, well, intuitively that works if fitness is perfectly additive, right? So fitness is just like the SAT, math plus verbal equals your total score. But if there is epistasis, and it's not quite like that, then in general, natural selection does not lead to negative LD. It might lead to positive LD or something else.
So I'm a little confused about that, because the parent-child, I thought that what matters for the parent-child inheritance is the additive genetic variance. I guess there's also an epistasis.
So that's very complicated. Pairwise and higher order interactions between additive effects at different sites do enter into the parent-offspring correlation, except that their contribution decays geometrically with the order of the interaction. But it's complicated because it turns out that in certain cases that those contributions can be almost exactly canceled by the generation of LD resulting from selections. So yes, we could have a whole seminar about this. Okay, any other questions? Okay, so these ideas and others have been developed mathematically for the last 100 years or so. But now, because we have all this new fancy technology now, it's possible to test these ideas experimentally. So a recent study of experimental evolution, this paper just came out in Nature. This has confirmed that sex does indeed accelerate adaptation through exactly this mechanism by attenuating the sign of negative LD. But this is arguably a rather special result. It's experiments on one organism, C. cerevisiae, and they're placed in these petri dishes that are artificially made extremely harsh for them. So how can we generalize this result to natural populations? Well, here's how we might do this in an ideal world. So my colleagues and I are currently working on a GWAS of number of children. At the current stage, the sample size is roughly 330,000 individuals. So this work may allow us to test whether SNPs associated with fitness, fertility in natural, but fertility is a pretty good proxy for fitness in modern humans because our mortality is so low. Or even better, if you had records of who's died since you performed the genotyping or whatever, that would be good too. So this is a way for us to see whether pairs of fitness-affecting sites in humans do indeed exhibit negative LD. So let's imagine that everything goes perfectly. The sample is perfectly representative, so there's no selection bias affecting it in a way that mimics natural selection. On the x-axis, you have the products of the GWAS regression coefficients signed so that the reference allele is the fertility-enhancing allele. The y-axis is the square root of R squared appropriately signed. Again, the reference alleles being the fitness-enhancing alleles. And so if we saw something like this, where LD is indeed negative and it becomes more negative as the effects on fitness increase, this would be a very, very, very nice result. All right, so don't put any stock in these numbers. I totally made these up. Although I do want to emphasize that in the real case, whatever these numbers are, they are going to be indeed extremely, extremely small. But I've seen that once you aggregate lots of SNPs in sample sizes in the hundreds of thousands, even when the axes have these minuscule scales, still you see these extremely regular patterns. So I'm reasonably optimistic that this is something that's feasible. So I should point out that a finding of positive LD does not necessarily rule out this class of hypotheses. So for example, positive LD, there might still be selection and drift or whatever the relevant mechanisms are that are producing the negative LD, but they might also be canceled by other things that are going on, such as, for example, assortative mating, which tends to lead to positive LD. But there's an asymmetry in that if we do find negative LD, and as I said, increasing in magnitude with the products of the effects on fitness, this can only mean that we do indeed adapt by natural selection more efficiently as a result of our sexual nature. All right, so there are various other details that GWAS and other types of studies can fill in. So I already mentioned some of these, such for example, what fraction of the genome is functional, and relatedly, how many deleterious de novo mutations does the average person carry? What is actually the heritability of fitness? So we have an idea about this from twin studies and GCTA and so on. But more can be done. And to make it all consistent, is it in fact the case that all of our observables are consistent with each other? Enough of the genome is functional, and you multiply out the mutation rates. There are enough deleterious mutations entering the population each generation to actually generate the heritability of fitness observed. And is this enough to overcome the twofold cost of sex? And all these other things I mentioned, such as what exactly is going on with epistasis. So it seems to me that this is a very exciting time to be interested in these questions. We may soon find a satisfactory answer to the enigma behind this most profound source of joy and misery in the human condition. Okay, so to summarize what I've talked about today, behavioral genetics studies the inheritance of psychological traits. Although it presents many possible interfaces with other scientific fields, it has for various reasons been historically isolated. But there's good news on the horizon, perhaps. The modern DNA, or maybe it's already here. The modern DNA era promises to give new life to the interdisciplinary reach of behavioral genetics. The rationale of sexual reproduction is one scientific puzzle that the new behavioral genetics may be able to address. Naively, this is the problem. It seems that asexuality should dominate sexuality by at least a factor of 2. And so this is known as the twofold cost of males. Now, a common theme of many hypotheses trying to account for this is that sex and recombination break up negative linkage disequilibrium between distinct fitness-affecting sites, allowing natural selection to eliminate deleterious alleles and promote beneficial ones. One of these hypotheses, although it's not widely accepted now, but anyway, one such hypothesis supposes that selection itself produces the negative LD. That is, the minus alleles hide among the plus alleles. And so therefore, sex can be desirable if it restores minus minus, which can be eliminated, and plus plus, which can increase further in frequency. There is now empirical support for this type of mechanism from experimental studies of evolution in model organisms, namely C. cerevisiae. Further support can potentially come from observational studies of natural populations. And Homo sapiens is the natural population that is the easiest to study of all. Try to ask 1 million butterflies or peacocks or whatever to herd into your study and donate DNA. A powerful piece of evidence would be a finding of negative LD between sites that affect fitness in humans. And we may soon be able to estimate both fitness effects and LD in samples exceeding 1 million individuals. An answer to the riddle of sexual reproduction would be a part of a powerful framework to explain sex differences in morphology and behavior that are observed throughout nature. It's my own view that no attempt to provide such explanations can be fully intellectually satisfying without knowing why there are two sexes, which is something I didn't even talk about today, or why many species must reproduce sexually at all. Okay, so that's everything I want to say. Thank you very much.